Thank you, Charlotte, Anna and Ed. That was incredibly inspiring, and I'm sure you'll agree, an amazing way to start our conference. So thank you again, very much indeed. So we'll continue this session now. This session is celebrating 150 years of Huntington research. The next two talks are about Huntingtin, the protein that goes right to the heart of this condition. We have one talk on mutant Huntingtin and one talk on wild-type Huntingtin. And our first speaker really needs very little introduction to this audience. Gillian Bates is a professor of molecular neuroscience at UCL. In her early career, she did work which contributed to the identification of the gene. She created the first transgenic Huntington model of Huntington's disease, which then led to identification of aggregates. And we're absolutely delighted, Jill, that you've come to talk to us today.
Well, thank you, Anne, and I'd like to thank Patrick and the organising committee for giving me this opportunity to tell you about some of our recent work, and also to echo all of the sentiments about how great it is to be back at an EHDN meeting. So I want to start by saying I think it's becoming more and more accepted that the very early events that at the molecular level that cause Huntington's disease is a two-step process. And step one is concerned with the size of the CAG repeat, so the length of the CAG repeat itself. So as you all know, the CAG is at the beginning of the Huntington's disease gene. And when we measure the size of that in blood, if you have 35 or less, you will not develop Huntington's disease. 40 or more within normal lifespan will lead to the disease. And then there's an increasing risk of developing the disease with CAG repeats of 36 to 39. But we've known for some time that the CAG repeat size that's measured in blood might not be the same as the CAG repeat that's in other cells, and that in certain cell types and certain tissues, the CAG can expand during lifetime, so it gets longer. And Peggy Shelbourne showed almost 20 years ago that in brain you can measure some very long repeats. And this phenomenon of increased CAG repeat expansion during the life course is called somatic CAG repeat expansion. And this here is an image from one of Peggy's 2003 paper, where she's showing this person who had 51 CAGs in their blood. Each of these little marks here is a single CAG in a brain cell, and you can see that she could see ones going up to several hundreds of kilobase pairs. And now Steve McCarroll's lab in Boston is using much more modern techniques to measure these in great detail at the single cell level. So we also know that the length of the CAG repeat that you measure in blood, to some extent, predicts when the disease might start in people. But it's not the only thing that predicts that. And so if you look at a given CAG repeat size, I think I'm probably pointing to something around 42 or so here, then you can see that the age of onset can vary dramatically from one person to another. And so there are other things that influence this. And we know that some of that is the genetic background that varies between different people. So some really insightful work over the last few years from Jim Gusella and Leslie Jones' labs and Sarah Tabrizi's labs has given us an idea of what some of these factors that influence this age of onset might be, called genetic modifiers. CAGs further expand from what's measured in blood that drives the age of onset of the disease and the disease progression. Now, We don't think it's the CAG in the DNA that causes the disease, so there has to be a step 2. And so the work I'm going to talk about now is what we think is one possibility of what this step 2 might be. So, as we said before, the CAG repeats at the beginning of the HD gene, and this contains what are called 67 exons interspersed with DNA called introns. And when that's turned into the messenger that goes on to make the protein, all of the introns get removed and you end up with the messenger. So if all of the introns are removed, you end up with a full-length messenger, which encodes the full-length protein. And in that protein, this CAG encodes for a polyglutamine residue. So we found a few years ago that it doesn't always quite happen like that, and that sometimes this production of the messenger terminates within this first intron, producing this very small messenger that just contains the exon with the CAG. And we know this produces something we call an exon 1 protein because it's the protein just made by exon 1. And we've known from 25 years' work that this exon 1 protein is very toxic. At low levels, it causes very rapid pathogenesis, and it's very prone to aggregation. And this is just a picture of showing you aggregated Huntington in nuclei and scattered between the nuclei and the cytoplasm of brain cells. So we're proposing that it's this, and so the important thing is the longer the CAG, the more of this termination in intron 1 occurs. So we're suggesting that this is a good candidate for the step 2 in the pathogenesis of Huntington's disease. So now I'll just go into a few more details for the next slides. So we're proposing that transcription terminates in intron 1 and activates cryptic poly-A sites. And this is the sequence of exon 1. This is intron 1. If exon 1 splices to exon 2, the first base of exon 2 completes a proline residue. If it doesn't splice, the first base of the splice donor completes the proline residue followed by a stop codon. So this exon 1 protein here that's made, it terminates in a proline residue and it does not contain any amino acids that are not present in the full-length protein. And we first showed that this was occurring in knock-in mice with David Hausman. This is showing you RNA sequencing across the Huntington's disease gene at the beginning of the gene in wild-type mice and mice that are homozygous knock-in mice with long CAGs. And here you can see in wild-type mice, you see reads over exon 1 and exon 2, but nothing in the intron. But if we look in the knock-in mice, we have these reads extending into the intron. And we now call, this is that little messenger, and we now call that Huntington 1A. The real name for it is Huntington 1A. And we know that in mice, the cryptic poly-A sites, there are two, one at about 680 and 1145 base pairs into the intron. And this occurs in all brain regions, all tissues. And we know this is an example of how this increases with CAG repeat length. This is data from Jim Rosinski at the CHDI Foundation, who has done a lot of RNA sequencing in knock-in mice with different CAG repeats. And this is looking at these intron 1 reads in the cortex. And you can see that as you go from 20 CAGs up to about 180, the amount of this increases. So the production of this small fragment increases with CAG repeat length. Now, we found that we have a very important resource for working with this, and that's an antibody called MW8. And it binds to the very end sequence of exon 1. Here you can see that proline residue. This is the end of exon 1 protein. And Christian showed a few years ago that this only binds to this sequence when it's at when that proline residue is at the end and it's exposed. And he showed that by deleting amino acids or adding amino acids, this would be in exon 2, making constructs, putting them in cells, expressing them, and then running a Western blot. And so this shows you constructs here. This is exon 1. We remove one amino acid, add one amino acid, etc. If you probe with 2B7, you see all of the constructs. Probe with S830, antibody to the whole of exon 1, you see all of them. Probe with MW8, you just see exon 1 protein. This is a very important resource for us. So now we can show that that Huntington 1A message is turned into the exon 1 protein in knock-in mice. So here, this is the knock-in mice again. We're immunoprecipitating, we're pulling down the protein with an antibody that just detects the mutant. So there's no signal in wild-type mice. This is the control lane. So we're pulling down all of the mutant Huntington, probe with S830, you see all of a pattern of bands, probe with MW8, you just see one band, that's exon 1, it ends in proline. And then probe with an antibody to exon 2, 3, there's nothing there. So that pattern is exon 1 protein. And here's another knock-in model, and we see it again. So Huntington 1A makes this exon 1 protein in knock-in mice. Does this occur in human tissue? So when Andreas Neuder was in the lab a few years ago, he used a qPCR to look for intron 1 in human brain. So in humans, intron 1 is 12 kb in length, and the cryptic poly-A sites are 2.7 and 7.3 kb into the intron. And here we're looking at somatosensory cortex, hippocampus, and cerebellum, and looking at the intron read depth again, if you like, and we see that in postmortem brain, it increases with CAG repeat length. So Huntington is alternatively processed to produce this little messenger RNA called Huntington 1A. The level of incomplete splicing increases with CAG repeat length. All knock-in mouse models make an exon 1 protein. We know that it's very aggregation prone and highly pathogenic, and we hypothesize that it's this exon 1 protein that initiates aggregation. And therefore, in order to look at this in more detail, we have to be able to track soluble and aggregated proteins in our tissues and in our cell lines. So I'm going to say very little about some of the work we've done on developing Huntington bioassays, but Christian Landels has a poster on this, and he will also be giving a short talk tomorrow where there'll be a lot more detail. So we started doing, just expanding on work that had already been done using combinations of antibodies to track soluble and aggregated protein. And Christian started working with 6 antibodies, 2B7, which is at the end of the exon, end of the protein. This one, MW1, just detects long polyQ, so only mutant. 4C9 is specific to a human sequence in exon 1 that you're going to hear a lot more about from Elena. MW8 is our antibody at the end of exon 1, and then we have some that are more C-terminal in the sequence. So he started off with 30 combinations of these in order to increase the number of assays that were available and build on the ones that were already out in the literature. He used 3 assay platforms, HTRF, AlphaLISA, and MSD, used a triage strategy to select the most useful antibody combinations, and then expanded this over the last couple of years to include more antibodies. So of course, what I'm going to show you, and is also on Christian's poster, is that we were most interested, of course, in whether MW8 might help us further detect and track exon 1. And so this is showing you that in soluble assays that track with soluble Huntington and aggregated Huntington, MW1 makes them exon 1 specific. So in order to show this, we used a mouse model that was made by Dave Ford-Shelton and Chris Ross many years ago, that it's transgenic for the first 3 exons of Huntington. So it doesn't contain any introns, and therefore you can't make that exon 1, you can't make Huntington 1A, you can't make the exon 1 protein. So if, first of all, we're looking at this is an assay that will just track all mutant Huntington. And here we're looking at ZQ175 mice and then these little transgenic mice with their wild type controls. So this is detecting mutants, so there's nothing in wild type. If we just can see that the soluble mutant Huntington decreases from 2 to 6 months of age in our knock-in mice, and it's also there in the cDNA mice and reduces from 3 to 6 months. If we use 2B7 to MW8, which we hope would be exon 1 specific, again, we see a reduction in our ZQ. We know that exon 1 is present in the knock-in mice, and there is no signal in the transgenic mice. So they don't produce exon 1, and this is a specific assay. But what really surprised us was that actually this also works for aggregated Huntington. So here's an assay that Christian developed that would detect all aggregated Huntington, whatever N-terminal fragment or full length was there, it would detect it. And here you can see it increases from 2 to 6 months in the knock-in mice, and it's also there in the transgenic mice. But when we use aggregation assays that use MW1, like 4C9 to MW8 or MW8 to 2B7, again we see aggregation increasing in the knock-in mice, but there's no signal in the transgenic mice. So it's also showing that it tracks with exon 1. It's showing you that it's exon 1 aggregation in these knock-in mice. So Christian has developed assays to, and complementing those that were already available, total mutant Huntington, that's exon 1 and full length together, soluble mutant full length, soluble exon 1 Huntington, total full length mutant and wild type, and a soluble mouse Huntington, aggregated Huntington and aggregated exon 1 Huntington. I'm not going to say any more about those except to say that the use of these can be quite complex, and so if you want to look more, learn more about the use of these assays, please go to Christian's poster. So what did we first apply them to? We're working with ZQ175 mice a lot. They're within a knock-in line within the field that's used a lot for preclinical testing and target validation. So we first thought we would see how quickly we can detect aggregation in these mice. So in the published literature, these mice don't really develop any phenotypes, like behavioral phenotypes, till 10 months of age or onwards, just by using standard techniques. So we first of all used, it's just showing you an aggregation assay. We looked in 10 CNS regions from these mice. This is our exon 1 aggregation assay. And this shows you in every region of these mouse brains, you can detect aggregation with this assay by 2 months of age. And the most aggregation is in the striatum. These can all be compared. They were all run at the same time, so these levels are comparable. What does this look like by immunohistochemistry? This is work, all of this work's described on a poster that Karupa's presenting here. Here we're looking at S830 immune staining. We have low power. We can see a staining increasing in the striatum and throughout the cortex as we go from 1 to 6 months. This is thresholding. Look at a higher power at 2 months of age. We can see a clear signal that's not present in wild-type mice. We were able to show that we could actually detect this as early as 6 weeks. Now, each of these signals is actually a cell nucleus. So how do we know that's aggregated protein? We can show it's aggregated protein by the following. This is an antibody from Steve Finkbeiner that detects the polyglutamine tract. So if you just use that antibody on a section from a ZQ175 mouse at any age, you'll get no signal because the polyglutamine is buried in the aggregates. So this is a 2-month section from ZQ175 mice. There's no signal. But if we pre-treat them with formic acid, we break open the bonds that are holding the glutamines together, and now the antibody can get to see those glutamines. So this is all 2 months of age. Here again, we see aggregation in the striatum, layer 4 of the cortex, and as we go to higher power, each of these are cell nuclei. While we haven't proven this, I would suspect that every medium spiny neuron in the striatum of ZQ mouse is full of aggregated Huntington at 2 months of age. So it occurs very, very early. And just using our set of assays for the striatum, these are the 3 aggregation assays. You can see that in 2 of them we can pick up aggregation at 1 month of age in these mice. This is our soluble exon 1 assay. So exon 1 Huntington is decreasing over that 6-month period, but if we look at total Huntington, this is measuring wild type and mutant together, there's no change. This is endogenous mouse. So it's soluble exon 1 Huntington that's decreasing over that time scale. So this is more support for our theory that it's the exon 1 protein that is initiating aggregation in these knock-in mice. So can we make mice that don't produce exon 1 in order to test this hypothesis further? So in order to try and do this, we tried to prevent the production of that Huntington 1A, or at least stop the production of that messenger from ending in intron 1. And to do that, we removed all of the cryptic poly-A sites from intron 1 from this particular knock-in mouse. And then there are a number of possibilities that could occur because we're retaining the splicing sequences that are important at the ends of the intron. We might get complete splicing to produce a full-length protein. We might not splice from exon 1 to exon 2, but there's no poly-A sites there now, so it can't terminate, and this would get destroyed by a process called nonsense-mediated decay. Because we shortened this, we might activate poly-A sites in intron 2, which actually we know we do, but this should not get exported from the nucleus. And we might actually make this small protein, exon 1, exon 2, activating poly-A sites. So we managed to obtain multiple founders on a wild-type allele, and we're able to show that this didn't affect the production of the full-length Huntington 1 transcript. And we produced one founder with a deletion on the mutant allele, and this had a CAG repeat size very similar to our knock-in mice. And if you want to look at this in more detail, you can talk to Kat on her poster here. I'm just going to skip to the protein work for this talk because of time. So this again is showing you these blots. So Christian is immunoprecipitating with 3B5H10 and then immunoprobing with different antibodies. We've got wild type, our knock-in mice, and then what we call our delta intron mice where we've got the deletion in the intron. And if we probe with an antibody that will detect exon all fragments, you can see we see a similar pattern, but the level of this small fragment is much reduced. If we probe with our MW8, we can see that we see exon 1 in the knock-in mice, and while there's trace amounts in the delta intron, we've very much reduced it. This is another CHDI antibody that also acts as a neoepitope antibody, and this is an immunoprecipitation and immunodetection to show you the full-length protein. So this is the wild-type allele at the bottom and the mutant allele at the top, which look very similar in these mice. So what effect does this have on aggregation? So here I'm showing you 9, 12, and 17 months of age, cortex, striatum, and we're probing with S830 again. There's no signal in wild-type mice. You can see in our knock-in mice, we see aggregation appearing in the cortex and in the striatum, but it's much, much slower in the mice with delta introns. Here we're showing you two regions of the hippocampus. Again, we see it increasing in our knock-in mice and much, much more slowly in the delta intron, and with the dentate gyrus of the hippocampus, we can really rarely see any signal at all. So when we run our assays through this, here again we now have wild-type, knock-in mice, delta intron mice. This is a mouse version of that aggregation assay that Christian developed that will detect any aggregation. And you can see that it's increasing over time in the knock-in mice. And we are detecting aggregation in the delta introns, but at a slower rate, consistent with the immunohistochemistry. Sorry, I'm too far away. This is an exon 1 aggregation. So this assay is just detecting exon 1 aggregation. We can see that there's, again, exon 1 aggregation in the knock-in model. And while we can see trace amounts of it here, it's much reduced in these mice. When we look at the soluble proteins, this is exon 1 soluble protein. That's decreasing over that time scale. We know that there's very little exon 1. We're not really picking it up with this assay, although we could see it on the Western blot in the delta introns. And when we look at full-length Huntington, that's not changing over that time scale. So this, again, supports our theory that it's exon 1 protein that is initiating this process, and exon 1 protein is present in all these knock-in mouse models. So everything I've shown you so far has been to do with the mouse Huntington gene and protein. And so in order to look at the human gene, we turned to the AT128 mice made by Michael Hayden's lab. And so first of all, we wanted to see whether or not we could detect Huntington 1A. And to do this, Sandra used probes qPCR assays within this, before this second poly-A site here, within an intron down here, and then across the 3' UTR for the full-length transcript. And again, we can see, so the black is showing you the intron 1 levels. Intron 1 levels are higher than other intronic sequences, and they don't really change from 2 to 12 months of age. So we can detect the Huntington 1A transcript in these mice. What happens when we run our assays? So this is looking at aggregation. Here the youngest we looked at was 3 months of age. This is cortex, striatum, hippocampus, and cerebellum. This is an exon 1 specific aggregation assay. We can see aggregated exon 1 by 3 months of age in these mice. When we look at soluble exon 1, it's soluble exon 1 that is decreasing. These are showing you some of the immunoprecipitations and Western blots. I won't labor this, but just to say here again, we're detecting exon 1 protein, and we see the reduction really occurring between 2 and 5 months, similar to what's coming up in our assay. And when we look at soluble mutant Huntington, there's no change over that time scale. So Huntington aggregates in the YAC128 mice contain exon 1 protein. But here we then also turned to a protocol called RNA-scope to look at the location of these transcripts. And here, Sandra used a probe to, again, the intron 1 to look for intron 1 sequence, the Huntington 1A, the full-length transcript, and other intronic sequences here, intron 66. So what she noticed was that if we look at full-length Huntington, we do see some signal in the cytoplasm, as you'd expect, because it gets transported for translation. But she saw these large foci, or clusters we now call them, in nuclei, in neuronal nuclei. And when you do a probe with the Huntington 1, the intron 1, we also see that it co-localizes with these large clusters, whereas this intron 66 is not doing so. So we identified in the, here showing hippocampus and cortex, but it's throughout the brain, that nuclear RNA clusters contain full-length Huntington and Huntington 1A in YAC128 mice. When we look at mouse Huntington, so now we're looking at, this is human Huntington, this is mouse Huntington. Here we see again these large foci in the nuclei. They do not contain mouse Huntington, so they don't recruit mouse Huntington. We wanted to see whether or not we could see them in our knock-in mice. So these are the ZQ175 mice again. Here now we're looking at mouse. Huntington. This would detect both mutant and wild type. Again, we can see most of the signals in the cytoplasm. Some of it's in the nucleus, but that would be a typical pattern that you would expect to see. And when we look with our intron 1 probes, we don't see these structures. So these large nuclear RNA clusters, they don't form in the ZQ1 mice. So ZQ mice contain very long CAGs, and they also contain human exon 1. So this is not being driven by a long CAG or a human exon 1. We think it's potentially being driven by the intronic interaction between Huntington and Huntington 1A transcripts. So Anastasia Kvorova's lab has done a lot of work on this. They've analyzed those clusters in great detail in both the YAC128 and the BAC models. And Anastasia spent quite some time looking to see whether she could see them in post-mortem brain. And she has a paper that's currently in press. This is using a chromogenic version of RNA-scope. And these are controls. This is a control probe. And you might probably say here, with a probe to the Huntington 1A transcript, she can see these large clusters in the striatum and cortex of post-mortem brain. So Huntington 1A is alternatively processed to generate mRNA in YAC128 mice. It's translated to produce the exon 1 protein. YAC128 brains contain aggregated exon 1. These two transcripts are retained as RNA clusters, and we hypothesize that it's intronic sequences that are keeping them there. So we know that if you have a long enough CAG repeat, you produce Huntington 1A transcript. And we know that this gets exploited to the cytoplasm for translation to the exon 1 protein. And we think that this is what triggers aggregation. And whether it does that will depend on a number of factors. The length of the glutamine, because the longer the glutamine, the lower the concentration. So aggregate formation is a concentration-dependent process. The level of the exon 1 protein, the shorter the fragment that polyQ is, the faster it will aggregate. So the level of this exon 1 protein, post-translational modifications, we'll hear from Hilal in a talk later today about how those affect aggregation, soluble localization interacting protein, and of course the protein folding homeostasis of the cell. And if the threshold concentration is reached, we would say that, do other fragments of Huntington contribute to this? Most likely, we think that they would get drawn in. So if, yeah, I'm almost there. If oligomeric and aggregated species are formed, and then we think that they form, sorry, I'm now pressing all the wrong things. They form, cause cellular dysfunction, which causes disease presentation and cause cell death. We also know that exon 1 Huntington goes back into the nucleus where it oligomerizes and forms aggregated species. That causes transcriptional dysregulation. But we now also know that Huntington 1A and full-length Huntington form these RNA clusters in the nucleus, and we don't know what the function of those might be. They could be pathogenic. These kinds of structures are pathogenic in other disease models like myotonic dystrophy, but they could also be protective in this case because they may prevent Huntington 1A being exported to the cytoplasm in order where it's translated to reach this threshold. And because this aggregation depends on a concentration threshold, it may be that somatic CAG repeat expansion is what gives us high enough levels to start that process. And of course, in cases where very long repeats are there already, like juvenile cases with ages of 1 to 2 or 3 years of age, you may not need that to occur. So finally, we propose that Huntington 1A and the exon 1 protein are effectors by which somatic CAG repeat expansion causes pathogenesis, and Huntington 1A is a therapeutic target. It provides a direct link between somatic instability and a pathogenic protein, provides a means of targeting the Huntington protein that's known to be highly pathogenic. It doesn't alter the levels of wild-type Huntington, and therefore is allele selective for all HD patients. And I'll leave you with a thank you slide for my collaborators and funders. Thank you.
Thank you, Jill. That